Hi, this is Miss Linton, and this is a 7th period review for Honors Bio, Unit 2. Say hi.
Hi.
All right. These are the top 10, but what I didn't put on my top 10, which I should have, would have been early Earth and life. So we'll start there, okay? But this would take you through chapters 1, 2, 3, and 4. These top 10 right here, okay? And then we just need to add on early Earth onto that. The other thing I want to remind you, okay, is you want, oh, that says Unit 1, sorry, but same thing. Know your labs. What was your big lab this time? The cell lab, right? So what did you learn? Sorry. So you had your cell lab and you looked at how to make a cheek cell, right? We looked at starch and iodine and glucose. Remember the whole setup with that? And then we had our eggs, right? And the way water moved through our eggs. Was that the only lab we did? That was it, right? And what?
The one that you did.
Yeah, the demo one with the starch and the iodine and glucose, right? You want to know your additional objectives for these different chapters, right? Be comfortable with your terms that are at the ends that were there that we kind of quizzes did. And then look over your group shared notes. Now, when you study, I know some of you still want that. When you study, you have input study and you have what? Output study. Examples of input study is reading your lab, because you still have the copy of the top of your lab, right? Reading your lab, reading your group shared notes, reading the answers to your additional objectives. Tonight, you're not going to read 4 chapters of textbook, and chapter 27. That's not happening, all right? But what's something you could read? The chapter summary. All of that type of work, that's all input. That's where you're reading and putting it in your brain. But where you really know if you can do it is when you do what kind of studying? Output. Now you know how this test is going to go. You're going to be working in a group for part of it. You've done this before. You're going to get 5 questions, right? And you know one of those 5 questions is going to be a what? Additional objective. And that could come from several different additional objectives, right? So one of your 5 is for sure going to be that. The remaining 4 questions are going to come from maybe a lab, which of course, you know you're going to have a lab question, you know you're going to have an additional objective question, and you're going to have 3 content level questions, right? Something about membranes, or something about the early Earth, something about the parts and structures of a cell, something about the characteristics of life, maybe the way something crosses a membrane, maybe something hyper or hypotonic. That will all be in the written part. Okay? And just like I did before, there will be 25 points wrapped up in that. Then part 2 will be your individual portion. And that will be all what? Multiple choice. There will be 40 questions, 2 points apiece, just like last time, is 80 points, right? 80 plus 25 is? 80 plus 25 is? 105. So you have 105 points. You have 105 points out there to get. That's what I call my curve. Or if I ask you a question and maybe you just didn't understand it, but you really knew the right answer, I just asked it in a weird way. That kind of covers our bases because I'm spotting you 5 before you even walk in. If you didn't show up for the test tomorrow, you'd have 5 points, right? I won't give it to you because you didn't show up. But, okay, there's 105 points right there, unless you're sick, of course. Okay, so there's 105 points out there for you to get. So if you know 80% of the test is going to be multiple choice type questions, then I would focus most of my reviewing on answering multiple choice type questions. Where could you find some multiple choice type questions? The quizzes would do that. What else?
The quizzes actually don't work on classroom.
I just re-updated some. If whatever ones I still need to give, I'll put them in. I'll put more in. Yes?
Questions at the end of the chapter.
Questions at the end of the chapter. Now, lo and behold, surprised the heck out of me. Somebody in my AP class, when I told, I said, you should, they go, oh, I do the questions at the end of the chapter. I go, do you grade them and analyze them? He goes, how do I grade them? You know where the answers are? The back of the book. Okay? What? Okay? Yes, at the back of the book. So what you're going to want to do is, I would personally, my strategy, because I'm assuming you have limited time tonight. You only have so many hours. Some of you have practice. Some of you have other things, or English or membeans you need to do, right? So you need to strategize on your time. One, focus on the labs. Make sure you know the lab. Read through the lab you wrote. Make sure you understand it. Two, you know you're going to get an additional objective question, so read through those so you're comfortable with them. If you choose to hyper-specialize in your team and you're like, you're doing chapter 27, you're doing chapter 1, you're doing chapter 2, you're doing chapter 3, you can do that, but you understand you're abdicating your learning, right? Because if they don't study, it's not their fault, it was your fault for asking them to do it for you. You understand that, right? Okay. Lab, additional objectives, and then you need to know your content. So the way I would approach it is I'd go, I wonder how much of this stuff I know stone cold pilot. What do I already know? So I would go in and do just all the odd questions for chapter 1, just the odds, and take it like a test. Don't sit there with your hand where the answers are and go, it's B or C. Oh yeah, B, I can see that. All right, yeah. I know, that one's definitely D. Oh, A. Oh, I didn't read that right. Okay, don't do it like that. Take out a piece of paper, take a pen, time yourself, put you like in testing situation, like you're like, there's 20 questions, I'm going to give myself 10 minutes, no more than 10 minutes. Set a timer on your phone, hit go, and hit it, just like recreate test conditions. Go through, do all the odds, take it like a test. When you're sure you're done, stop, go to the back, grade it. If you get an 85% on it, move on. Come back to it later, go do the next chapter. If you're not getting 85% or higher on it, go back, reread the group shared notes, reread the summary, take it again. But this time, instead of doing the odds, take the evens. See if you do better. Does that make sense to you? And go back and do some quizzes or something like that. So strategize how you use your time on this, so you get the most of your studying time. 80% of it's going to be multiple choice. Study like that. Spend 80% of your time taking multiple choice questions, right? Okay. Now, topics you need to make sure you know. I didn't put it up here, but on the early Earth and life, if you look at that, do you think I want you to know the parts of the Earth? What are the parts of the Earth? Core, you learned that last year with me, right? Core, then what comes next? Mantle, crust. What's making the crust move around? Convection currents in the mantle, right? Those kind of things. How old do you think the Earth is? 4.6 billion years old, right? How old is the first living thing? About 3.5 billion years old. I taught you that, right? It all is just an amalgamation in my brain. Okay. We did plate boundaries, right? So if the plates are moving around, you have like 3 options, right? They can go towards each other. What kind of boundary is that? Convergent. Then whatever one is bigger is going to go on top or less dense, and the other one has to go under, right? So if it's a continent and an oceanic plate, then the oceanic plate's going to go underneath the continent, right? And it's going to maybe come up as a what? Volcano, right? If it's 2 oceanic plates, one will go under the other. Whichever one is more dense will go under the less dense one. Yes? Okay. If it's not a convergent boundary, what's another kind? Divergent. Divergent. Sorry. Divergent boundary, where are you going to see that? Maybe it's seafloor, what? Spreading. There's a continent where that's happening as well. Right? And then what's the other one? Transform. And on transform, they're trying to slide past each other, but they usually don't slide past that well, right? And where you have that, you have fault lines, and you have what? Earthquakes. Where you have convergent boundaries, you tend to have volcanoes, right? Okay, so that's that first part. That was plate tectonics. We looked at evidence. We looked at earthquake locations. We looked at volcanoes as evidence. We looked at how where there's heavy bodies of water and ice, it creates like divots. Do you remember that? We looked at the reverse magnetism of the oceans. Do you remember all of that? And then we spent a great deal of time just talking about the oxygen, what? Revolution. Because that changed everything. Because the early Earth had no oxygen, right? No oxygen on the early Earth. It wasn't until non-cyclic photophosphorylation evolved by cyanobacteria about 2.3 billion years ago. But that cyanobacteria started generating a bunch of oxygen, which allowed organisms to be multicellular and come out of the water, things like that. We'll learn more about that later, but that was the oxygen revolution. Then the last part we learned about is how, remember how we said atoms build what? Molecules, organelles, cells. So we were talking about how that could have come about. And if you remember, early Earth was missing all oxygen, right? But there were molecules, but these molecules probably came out of what? Volcanoes. At what kind of boundaries do you think? Convergent boundaries, exactly. So now you had these gases on early Earth, but we had forms of energy too, right? What were the forms of energy we had? UV, radiation. What else did we have? Lightning, molten magma, radiation from the Earth's core. And that energy could have taken those gases that were devoid of oxygen and made simple organic molecules. And you remember how we said Operin was the guy who came up with the experiment, but Miller's the idea, but Miller's the one who did it. And he was like in less than 2 weeks able to form organic molecules. And then those organic molecules with that same energy could have been joined together to form large macromolecules that we now know as carbohydrates, lipids, proteins, and nucleic acids. You start putting those together and you can build cells. The first cell, was it a prokaryote or a eukaryote, do you think? Prokaryote, right? No membrane enclosed organelle, small. And then later, a eukaryotic cell could have evolved. Remember how we learned about the endosymbiotic hypothesis? Now, one prokaryote went to live inside of another prokaryote. Okay? And then that became the eukaryote. And we have evidence for that because we looked at mitochondria and chloroplasts. They have their own DNA, circular plasmids, their own ribosomes, and they're self-replicating. And I know that was a lot, but I wanted to hit everything on that part, in that early part of chapter 27, because that was a long time ago. So go back and skim over your notes. When you take the practice quiz on chapter 27, it only goes up through, I think it's 27.1. So when you take your practice test, don't stress out, stop where it's, once you've built an early cell, then don't answer any more questions after that, right? Because we're going to learn the rest of chapter 27 later. I don't want you to look at that and go, we never talked about that, because you're right, we didn't. I just talked a little bit about it, and I reviewed some plate tectonics from your 9th grade year. Okay, now here. Okay, when we looked at chapter 1, right, at the very beginning, the first thing we said was biology was the study of life, and we talked about the characteristics of life. Let's do the characteristics together. Ready? Here we go. Living things are organized, made of cells. They acquire materials and energy. They maintain homeostasis. They respond to stimuli and reproduce. They grow and develop, and they adapt and die. What is an adaptation of birds? What do they have? Wings, feathers, hollow bones. Those are adaptations. What is an adaptation of a cheetah? Speed. Sharp teeth, right? Those are adaptations. What is it when I get hot, I sweat, and I shiver when I'm cold? What am I doing there? Maintaining homeostasis. I want my cells to stay the same even though my environment changed. When I made babies, and I made 2 of them, what was I doing there? Reproducing. When I fed them, they were what? Growing and developing. Exactly. So when you think about the characteristics of life, right, think what are examples of those characteristics of life. The very first characteristic we talked about is living things are organized. So we discussed organization. It goes atom, then what? Molecule, organelle. Do all cells have organelles? Who does? Good. Organelle, cells, tissues, organs, organ systems, organism, and then here's unit 1, population, community, ecosystem, biosphere. I don't know what word I was saying there. Good. Perfect. Then we started talking about how organisms are organized. Do you remember the 3 domains? Bacteria and archaea and eukarya. What do bacteria and archaea have in common? They are both what? Prokaryotic. But they're different because archaea are the extremophiles, right? Those are the ones that live like where it's really salty or it's a high temperature or pH is really low. But bacteria are found everywhere. Neither one of these guys have anything other than cell membranes and ribosomes. They have DNA in them and they have membranes inside of them, but not organized into those organelles that we know and love and we have studied, right? Do you think it would be important to know your organelle structure and function? Yes. Who's the control center of the cell? Who does photosynthesis? Chloroplast. Who processes, packages, and distributes? Good. Where does protein synthesis take place? What's the stomach of the cell? Who decides who goes in and out? Who is supportive and protective and gases can go through it? Who looks like a churro? Who doesn't have centrioles? Plant cells. What do plant cells have that animal cells don't have? And? Chloroplast and cell wall. Good. Who's like a ribbon of transportation through the cell? It can be or endoplasmic reticulum, and it can be what? Rough or smooth. What makes the rough one rough? Ribosomes. Good. Okay. Good. Okay. Next, we did that. Remember we talked about the scientific method? Do you remember that? Right? We talked about making observations and asking questions. All your labs have driving questions. We do a little bit of research. That's like your background on your labs. Then we form a what? Hypothesis, an educated guess. Then we do a experiment. In our experiment, we always have a basis of comparison. Who's our basis of comparison? Control. Then we keep everything the same except one thing. Like if we were seeing how sugar water affects plant growth, we'd use the same kind of plant. We use the same amount of water. We just vary the amount of sugar. What do you call the varying the amount of sugar? Independent variable. How high our plant grows is the dependent variable. Because we let them have the same light, same sunshine, same amount of fluid. Those are called what? We're keeping it constant. Who would be a good control plant in that scenario? How about just plain water? Does that make sense? That's our basis of comparison if we're doing sugar water on a plant. Now, I know I'm talking fast, okay? But the reason is I'm trying to hit a bunch of things, so at some point you go, oh, wait, I better review that. Oh, wait, I better review that, right? You're not sitting here highlighting everything I say, right? All right, okay. Okay, once you do your experiment, then you analyze your data, maybe with a chi-square test. I don't know. We've done that before, right? Observed minus expected squared over what's expected, sum it up, look on the table. And then we make our conclusion, claim, evidence, reason, yeah? Okay, I did that. Oh, then we got into chemistry. So let me go back up here to my, okay? So we did characterization, organization. Okay, so let's get in the atom story. Atoms are made of protons, neutrons, and electrons. The protons and the neutrons are in the nucleus, atomic mass. The electrons are in energy levels. 2, 8, 1832. 8, 8, 8 is great octet rule. The number of protons is called the what? If I vary the number of neutrons, I'm talking about an isotope. If I vary the number of electrons, I'm talking about a ion. What makes an atom happy? Why does it interact? It's trying to get stable and happy. How can you get it happy? Fill its outermost energy level or get it to 8. So, I'm not happy by myself. I have to get it with somebody else. What are the different ways I can do that? I can form a bond or bond. What is that? Ionic or covalent. In ionic, I'm ditching some electrons to get stable. But if I ditch electrons, what charge will I become? Positive. If I take somebody's electrons to fill up my outermost energy level, I will become negative. What do opposites do? Attract. That's one way you form bonds. Atoms, molecules. I'm telling you how you form molecules from atoms. Ionic bond. What's the other bond? Covalent. When you're in a covalent bond, what are you doing? Sharing. If I share fair, everybody's got the same number of electrons, so nobody's got any more than anybody else. That's called a what kind of bond? Nonpolar covalent. If I share unfair, who is the classic unfair sharer? Oxygen. Who does he normally take them from? Hydrogen. So when he shares unfair in his polar covalent bond, he becomes slightly, what's his charge? Negative. And the hydrogen he's hooking up with in water becomes slightly positive. So this lends itself to another kind of bond. What kind of bond is that? Hydrogen bonds. Okay? And in hydrogen bonds, if you remember, if you have okay, what is the name of this bond right there? What kind of? Polar covalent, right? But this bond right here, what bond is that bond? That right there is an H bond, okay? And it's really a little bit too heavy, okay? But it would be more like, right? All the pink ones are hydrogen bonds. And that gives water its property. Like it has high heat capacity, high heat of vaporization, adhesion, tension, cohesion, right? Now, any kind of anxiety you are feeling right now, let's learn a lesson from this anxiety you may have right now, because we can't change. You either have reviewed all along or you haven't. It would lessen your anxiety if you would review every chapter all the time, right? Let's remember that. Let's remember that in this moment where we're going, damn, we talked about a lot this unit. I forgot all about that, okay? That's why I say review a little bio every day, input and output studying. It puts you in such a better spot. Okay, you can't cry over spilled milk. Just let it sour and let's move on, okay? Now, we talked about the properties of water. So if you look back at our list, ionic, covalent, hydrogen, pH. pH, remember pH? It's the number of hydrogen ions, right? The low number, because it's in inverse log, the low pH is a lot of hydrogen ions and it's a what? Acidic. When you have few hydrogen ions and you have a lot of hydroxide ions, it's not an acid, it's a base. Okay? pH. What's neutral? Water. And what number is that? 7. Is rainwater neutral? No. Okay. All right. So we did 1, 2, 3, and we did early Earth 4, organic molecules. All right. What are the 4 important organic molecules? Carbohydrates, proteins, lipids, and nucleic acids. When you want to join monomers to make polymers, what is that reaction called? Dehydration synthesis. Another name? Condensation synthesis. When you want to break it apart, what is it called? Hydrolysis. Good. So what is the monomer? You know you have 4 polymers. So here's dehydration synthesis and hydrolysis. So carbohydrates. What's the monomer of all carbs? Sugars. Good. Remember how we learned about the different kinds of sugars? Sucrose, fructose, and galactose. Glucose plus glucose. Wait, did I just say sucrose? Okay, good. Glucose, fructose, and galactose. I didn't know what I was saying in my head. Okay, so maltose is glucose plus glucose, right? Fructose is glucose plus, sucrose is glucose plus fructose, and maltose is glucose, and lactose is glucose plus galactose. Right, okay. And what is a complex carb we talked about? In us, how do we store our carbs? Glycogen in our liver. Plants can store it as what? Starch. Or they can build stuff with it like cell walls, like cellulose, right? Or chitin, if you're, chitin like fungal cell wall, right? Okay, what's another important organic molecule? Do you have a chart that summarizes all of this? Yes. What's another one? Carbs, then what's another one we learned? Lipids, but there's two kinds of lipids, right? What are the two? Fats and steroids. Fats is glycerol with fatty acid chains. They could either be what? Saturated or unsaturated. Saturated ones are found in animals, solid at room temperature. Plants have unsaturated liquid at room temperature. Steroids are things like cholesterol, right? Testosterone, estrogen. Those are steroids, and those are 4 rings of carbon joined together. Okay, give me another important molecule. Proteins. Proteins are built out of what? Amino acids. How many different amino acids are there? 20. Now give me that last one. Nucleic acids. What are examples of nucleic acids? DNA, RNA, and what? ATP. All right, we have a whole chart. You can remember those. We did dehydration, synthesis, hydrolysis already. We reviewed prokaryotic and eukaryotic cells. We did plant versus animal. Now we're in structure of cell membranes. That's what we just learned. Phospholipids. What else are in cell membranes? Carbohydrate side chains, glycolipids and glycoproteins. What else? Steroids, the cholesterol, and what else? Proteins. How do I get through a cell membrane? I can go through the phospholipid bilayer. I can use a channel or a carrier or whole membrane, endo and exocytosis. Phospholipids create a barrier. Cholesterol helps with fluidity, keeps it together. Glycolipids, glycoproteins are for what? Cell recognition, our fingerprint, right? Proteins, let's review proteins. There are, breathe. This is just a test. It's one test your whole life. One test, you're like, I can't even remember all the tests I've taken in my life. Am I doing just fine? Yeah, so will you. So will you. You're going to do the best you can with the time you have. You can't review a month's worth of lectures in 45 minutes. I'm attempting to, right? I'm attempting to. I'm dragging your asses through. Oh, sorry. Right? I'm reviewing all of it, and some of you are like, okay. I know that. So you're going to pick out the parts that's the hardest part and study those more, right? You're going to focus on taking those questions at the end of the chapters, right? It's not the end of the world. It's just a test. There'll be easy parts. There'll be hard parts. You're going to do just fine. Let's talk about proteins, okay? I love you. I didn't write an obnoxious test. I just have some things I need you to know. Okay, here you go. Too many things, Ms. Lynn. Here we go. Channels. What are you doing right here? Here we go. Channels, carriers, cell recognition, receptors, enzymatic, and junction. Okay? Then we already did ways to cross the membrane, right? We already did that. Active versus passive. Active requires what? Energy. Passive does not. Let's talk about through the phospholipid bilayer. If you're going through the phospholipids, it is always passive because you're going to go from a what? Higher concentration to a lower concentration, right? When water does that, what is it called? Osmosis. And then we say either side of the membrane, it's either hypertonic, hypotonic, or isotonic. Isotonic is same. Hypertonic, you have more what? Solute or solvent when it's hypertonic? Solute. More stuff. You put the sugar in your coffee. Solute. You put the sugar in your coffee. Hypertonic, high sugar. You put it in there, solute. Okay? Hypertonic, hypotonic. Water, you have more water in a hypotonic solution. Remember, water must flow from the hypo, going from a higher concentration to a lower. If you plop a cell in a hypotonic solution, what will happen to that cell? It'll get what? Bigger. If it's a plant cell, where will it go? Vacuole, large central vacuole, right? If you plop a cell into a hypertonic solution, what will happen? The cell will get smaller. What will keep it same? Isotonic. Good. All right? That's all passive. What's another kind of passive transport? Facilitated transport. Facilitated transport uses what? Channels or carriers when you're going from a high concentration to a low. If you're going to go from low to high, that's going to cost you some energy. And when you use a protein to do that, it's called active transport, and that protein's called a what? Pump. Good. When you use a whole membrane and you're moving cytoskeleton around and you're doing endocytosis or exocytosis, do you think that's going to cost you energy? Yes, that is also active. Let's talk about the different kinds of endocytosis. What do you call cellular drinking when you engulf fluids? Endocytosis. Cell eating? Phagocytosis. Then what's the other one where it binds, and then it does this? Receptor-mediated endocytosis. All the exocytosis is same. Okay, we did it all. Just like that. It only took us, did I record it?
Oh, God.
I did. 31 minutes. It wasn't moving. Were you worried?
Very.
A little bit?
Too much.
Yeah, I knew we wouldn't have time for all the slides. That's a few
What about you share them with us?
What?
Let's share them with us.
Yeah, what?
I have no problem doing that. Yeah, these are not like set up. They're the old way.
Oh, yeah.
So you can't see them. It's super annoying. Sorry about that. All right. Are there any questions you want to ask me? Any questions you want to ask me? Okay, so what did I tell you? Best way to study? Test. So 80% is going to be multiple choice. 80% of your time. Go hit the questions cold. Chapters 1, 2, 3, and 4, and the beginning of chapter 27. Hit them cold. Only take the what? Odds. Grade it. Analyze it. Study what you didn't know, and then you can go back and take the evens. If you did really poorly on one of those chapters, then go back and study your notes, right? Look at the diagrams, read the summary. You can eat that in here. And then also make sure you know your additional objectives and what else? Your labs. You can do that, right? I know you can do that. Okay, you're super smart. I'm already proud of you. Make good choices. That's it. Good job.